I am Dr. Nikita Sharma, Associate Professor Chemistry, SPNKS Government PG College, Dausa. Today we will start enzyme model, host guest chemistry. Most people are familiar with the lock and key theory of enzyme activity. According to this theory, the catalytic activity of enzymes stems from the fact that the substrate, that is, the compound undergoing reaction, fits tightly into a pocket in the surface of the enzyme, much like a key fits into a lock. Once inside this pocket, the substrate is held in close proximity to a reactant which converts it to product. The activity of enzymes has served as a source of inspiration for the research efforts of many organic chemists. The enzyme chymotrypsin has played a central role in the research of Nobel laureate Donald J. Cram, who for 40 years has been trying to answer the question, what makes enzymes such good catalysts? Professor Cram's attempts to answer this question have led to the development of an entire field of chemistry known as host-guest chemistry. This topic provides an overview of that field and serves as our introduction to chemical reaction. We will start with a brief survey of one class of reactions, nucleophilic substitutions. Equation 1 describes a reaction that everyone is familiar with, the neutralization of hydrochloric acid with sodium hydroxide. To an organic chemist, reaction 1 is an example of a substitution reaction. The chlorine atom of the hydrogen chloride is replaced by the hydroxy group from the sodium hydroxide. The color coding is included to mark this reaction theme apparent in equation 2 to 5. Equation 2 depicts a process called a nucleophilic aliphatic substitution reaction. In this case, the hydroxy group replaces a bromide from the methyl carbon atom. We will consider this reaction in detail when we examine its mechanism. Our next example illustrates a process known as saponification. Here, the hydroxy group substitute for an OR group of an ester. This reaction is called a nucleophilic acyl substitution. Equation 4 provides another example of a nucleophilic acyl substitution reaction. In this case, the functional group involved is an amide instead of an ester. One more for you to digest before we move on. Reaction 5 describes the hydrolysis of a protein, a polymeric form of an amide. This is precisely the type of reaction that chymotrypsin catalyzes. Chymotrypsin is a member of a class of enzyme known as serine protease. This type of enzyme catalyzes the hydrolysis of protein, that is digestion. The active site of all serine proteases contain 4 common elements. First is a binding site. Second is an hydroxy group, that is a part of a serine residue in the polypeptide chain. Third is an imidazole ring, that is a part of a histidine residue. And fourth is an aspartate group, that is a part of an aspartic acid residue. As you can see from the model of chymotrypsin, there is a lot of the polypeptide that is removed from the active site. This fact led Professor Cram to ask a more specific question, namely, what are the minimum structural features required to observe chymotrypsin-like catalytic activity? In order to answer this question, Cram and his colleagues spent hundreds of hours building CPK models of potential complexes and grading them for desirability as research target. The final target they selected as a host is shown in figure 1. This is figure 1. It contains all 4 of the elements found in serine proteases. That portion of the structure shown in purple in the binding site. Purple in the binding site. The ARCH2OH group shown in red is an analog of the serine residue. Red. The imidazole ring shown in blue corresponds to the histidine residue. Blue color shown the histidine residue. And the CO2 group is in orange fills the role of the aspartate group. CO2 group is shown in the aspartate group. This is the figure 1. The compound they selected as a guest and the reaction they expected it to undergo are illustrated in equation 6. This is equation 6. In equation 6, the HOCH2AR represents that portion of the host designed as a serine analog. In fact, Cram's group was not successful in their attempts to synthesize the host target. They did manage to prepare a related structure, however, and to demonstrate that it displayed remarkable catalytic activity. Figure 2 shows the structure of the product of reaction 6 when HOCH2AR represents the alternative host. This is figure 2. Note how the NH3 plus group is complexed to the binding site of this modified host. Presumably, the Coulombic attractions between the positive charge on the nitrogen atom and the lone pair of electrons on the oxygen atoms holds the guest in place so that the reaction 6 may occur more readily. The rate of reaction 6 when HOCH2AR represents alternative host is 10 raised to power 11 times faster than when HOCH2AR represents the model compound shown in figure shown in equation 7. This is equation 7.